Hi friends, in this we are going to discuss about the difference between ISOZYMEs, RFLP, RAPD, AFLP, SSR, SNP. And coming to the first one, abundance. The markers, ISOZYME markers are very low. It's contain a low markers. RFLP we contain medium markers. Medium means medium number of markers. And RAPD we will have very high number of RAPD markers and also AFLP markers. SSRs are high and SNP are very high. SNP single nucleotide polymorphism and SSR simple sequence repeats and RAPD rapid amplified polymorphic DNA, amplified fragment length polymorphism, and these are all the full form of those things and types of polymorphism. And the amino acid change in peptide, by this type there will be a change polymorphism we can see from one line to other line or one genotype to other genotype while doing gel run. And coming to the RFLP, how the polymorphism occurs means single base change, insertion, deletion or inversion. By this process there will be a polymorphism occurs with respect to RFLP. Same with respect to RAPD and AFLP. And SSR, how we are going to see polymorphism means this is alteration from one genotype to other genotype, polymorphism. And in SSR there will be a polymorphism due to repeated length variation. It means in this there will be a repeat length variation, like some of the repeats may 2 times, maybe repeated 3 times, repeated 4 times, repeated, like AU is repeated 3 times, sorry, AT repeated 3 times, 4 times, like ATG is repeated 4 times, 5 times, like this. There will be repeat in length variation. And single nucleotide polymorphism means a single base change. There will be a change in the single base, like if some ATG will be there, it's some others T G. There will be change in single base pair. And coming to quality of DNA, in this isosome protein markers there is no need of DNA, and in RFLP we need high, and AFLP also we need high, and all these three we need minimum quality of DNA. Means if we do want to do any experiments on RFLP, AFLP, the quality of the DNA should be very high. And sequence information for ISOZYME, there is no such things, not applicable. And RFLP, no need of DNA information, sequence DNA information. And RAPD not required, and AFLP also not required. In SSR and SNP because it is required. Most important. If you want to see any repeated length, we should check the SSRs to see the SSRs. Then we should know that sequence. By that only we can say that how many times repeated, by seeing those sequences. And also single base change, as I said here, if you know the sequence also, A is replaced by T or G. If you want to know previous one, we should have the previous information about the DNA sequence of that genotype. Then only we can find the SSRs and SNPs. And inheritance, it's a codominance. ISOZYMEs, RFLP codominance and its dominance, and AFLP alpha dominance. These only keep this. Remember that RAPD, AFLP are dominant. Means in this through these two markers we cannot differentiate heterozygous and homozygous. We cannot differentiate like this. We will get only single band with respect, but in codominance we can differentiate heterozygous and homozygous dominance conditions, because we'll get two bands, we can easily differentiate, but in this we will get only single band if it is a dominant one. By that we cannot be able to differentiate. For this, how to differentiate codominant and dominant markers, please refer our previous video. Reproducibility, ISOZYME is medium, RFLP is high, and SSR and SNP are very high, and RAPD is very low. Reproducibility means that if we done again, we should get same results. If we done that experiment again with respect to these markers, we should get same results. That's called reproducibility. So SNP, SSR and RFLP have high reproducibility. Coming to the technical complexity, and the isozymes are medium has less complexity, medium complexity, and RFLP is high, because in this we are going to use radioactive substances, though that's why it's somewhat it requires longer length and highly complex structure, complex procedure to do such things. So it's a very high complexity. Technical know-how should be there, and skilled person must be there. Highly skilled must be there. It requires more time, consumes more time. That's why. And these are low. This is low and medium, low and medium. Developmental cost. To develop these markers, it requires medium cost. RFLP require high cost, and it's high technical complexity also requires more. And this RAPD very low for reproducibility and low technical complexity, and developmental cost also less. And in this SSR is high because it's a next generation sequencing. Requires sequence information. These all required, though technically it requires more money cost. Species transferability, how to try from species to species. Species transferability is very high with respect to isozymes. RFLP is medium, and RAPD is high and medium, and it's a low transferability in SNPs. Automation means without doing much of the human work, we can differentiate easily. The machine should do itself every required processes. That's why this is called automation. Automatically it has to do all these processes, but as low automation, it requires much of a human work, and it's in RFLP as a low, and this RAPD is medium, means we can do some of the works automation, and same as RFLP, AFLP. But with respect to SSR and SNP, automation is very high. We can get results easily without much of a human work. Automation, my machine could do all those steps. And thank you. Please subscribe our channel for further videos. Thank you.